So our last lecture here on photosynthesis is pretty cool because we're going to talk about regulation, but what's really neat about regulation of photosynthesis is not only do we need to think about regulation of the process, there's a time sensitivity to this regulation. And as we'll learn about in the very last section, there's actually a spatial kind of aspect to this regulation with certain kinds of plants. So again, just to kind of highlight, the Calvin cycle is a way that a plant is able to use ATP and NADPH that are produced in the light reactions to fix carbon dioxide and make starches through the dark reactions. So again, that ATP can be used to make those starches, or that ATP can just be used for energy needs within the cell. Bottom line is when it's light out, the Calvin cycle needs to be active. However, when it's dark out, we don't want the Calvin cycle to be active because that's going to use NADPH and ATP as well. And the only way to make ATP and NADPH when it's dark out for plants is to do the same thing that we do. Go through a glycolysis, the citric acid cycle, and oxidative phosphorylation. So plants behave very much like us metabolically at night, is they need to use their reserves, catabolize their starch reserves, and use that in the same way that we use starches to make energy. But clearly we don't want to create this futile cycle where the Calvin cycle then is going to take that ATP and the NADPH from the pentose phosphate pathway and use it to remake the carbohydrates that it just broke down. So again, we need to make sure that we have the Calvin cycle not working when it's dark out. Again, as we'll see, the stroma basically contains all of the enzymes for these processes. So again, we're going to talk about how this regulates in a light-sensitive way. So again, we're going to do the same type of analysis we do when we try to identify flux control points. We look for far equilibrium reactions, so large negative delta G values. And if we look at them over here, we're going to see that there's three spots. We're going to have ribulose bisphosphate carboxylase, so not surprisingly, Rubisco is an enzyme that we'll see as part of the regulation process. But importantly, we're going to see two other enzymes, fructose bisphosphatase, or FPBase, and then the said heptulose bisphosphatase, or SBPase. So two phosphatase reactions that we're going to see that are important flux control points. And as we'll see in just a second, the activities of all three of these are going to be variable depending on the level of illumination. So again, it's all about Rubisco, all about the ability of this enzyme to be active and then participating in the dark reactions. So there's three ways that we really think about regulating this enzyme. We think about it from a pH standpoint, so that's not unlike we saw with activity with mitochondria, and we talked about the IF1 protein. That inhibitor. We also have magnesium levels that can control the activity of Rubisco. And then we also have an inhibitor, a transition state analog. So again, in our activity, we'll make sure to review a lot with mitochondria as we think about comparing and contrasting activity within the mitochondrion, and it's all about cytochrome C and what's in its reduced state for mitochondrion. And again, we'll think about how we have some parallels here with what we see with chloroplasts. So again, to highlight here, pH, right? pH optimum of Rubisco is going to be near 8. So again, when there's a lot of electron transport going on, means there's a lot of light out. We're going to have the pH of the stroma is going to increase from about 7 to at least 8. Remember, we talked about the difference, that delta pH that we have in chloroplasts is quite large. But as the pH rises, that's going to activate Rubisco. So again, coupling those two processes, the light and the dark reactions. Stimulated by magnesium. Remember, when we talk about as protons get pumped out to the inner membrane space, magnesium is going to come into the stroma because we want to equalize that charge. And so magnesium is also going to activate Rubisco within the stroma. And then lastly, kind of as a fail-safe sort of mechanism, we have a transition state analog that's going to be synthesized when it's dark out. So it ensures that if for whatever reason Rubisco decides it wants to try to be active when it's dark out, this transition state analog is synthesized. So it's an inhibitor, and it's again only synthesized when it's dark out. So to think just a little bit about how light kind of affects things. So that's thinking about Rubisco, right? So Rubisco here is that one flux control point. When we think about these two phosphatases, those two phosphatases are regulated by light levels. So what we really see here, much in the way that we think about phosphorylation cascades, we think about kind of redox cascades that are affected by light levels. So for example, if there's a lot of photons coming in and activating electron transport, when we have photosystem 1 kind of passing its electrons along to ferredoxin, that ferredoxin, in addition to having those electrons go to FNR or back to Q to participate again in the Q cycle, those electrons can also go to oxidize, or in this case reduce, this ferredoxin thioredoxin reductase enzyme, and then again that will go and reduce thioredoxin, and when thioredoxin is in a reduced state, it will reduce and activate this bisphosphatase. So again, a way to couple, there's light out here, let's make sure that our Calvin cycle enzymes are active. So again, we'll walk through this with an activity and also kind of highlight the important aspect that when we have this thioredoxin in this reduced state, this thioredoxin is also capable of deactivating PFK, so ensuring that glycolysis is not going on when there's lots of light out. So you don't need to be breaking down your starches to make ATP when you've got light out that can allow you to do it. So again, a couple of summary slides that just allow us to think about what it means when there's high levels of illumination, and again, ways to think about what's going on, whether or not it's day or night. So again, we'll have some activity questions that allow us to summarize that. The last thing I kind of want to talk about is this concept of photorespiration. We've already learned about the chemistry that Rubisco can do. So I'm going to flip to the next slide here for just a second. We talk about Rubisco basically taking, and it takes Rubisco, it adds a CO2 to it, and then does that cleavage reaction to give us 2 molecules of 3PG, and then goes through the Calvin cycle to generate GAP and then regenerate ribulose bisphosphate. So this is the pathway that we've already seen in the Calvin cycle. So what happens in photorespiration is they actually will use oxygen rather than carbon dioxide as a substrate, and so they'll consume oxygen and evolve CO2. And again, this is going to happen when there's low concentrations of CO2 and oxygen levels are high, and that's going to happen when there's a lot of light out, and we'll see that in just a minute here. But again, what happens here is this ribulose bisphosphate is going to use oxygen as a substrate with the same enzyme, and now it generates a different species because we're not adding another carbon atom, we're just adding oxygen. And so we're going to not only generate 1 molecule 3PG, but we'll generate this 2-phosphoglycolate. And so when we break this apart in an activity, what we're going to see is a lot of parallels to some things we saw, for example, with the glyoxysome. You're going to have a specialized organelle that's going to allow you to do this kind of chemistry. And it's called a peroxisome. So we'll get to that in just a second here. But just to kind of highlight, we'll spend some time in our activity going through reviewing this same mechanism. So again, it says to see this carbon dioxide mechanism, but look at how things are different when you have oxygen as a substrate rather than carbon dioxide. Again, we'll spend some time just kind of reviewing here CO2 as a substrate. And again, here is O2 as a substrate. So carboxylase chemistry, oxygenase chemistry. Carboxylase chemistry we see with the Calvin cycle, that's going to happen when it's light out. Oxygenase chemistry, that's going to happen when CO2 levels are depleted. So again, this is still going to be something that happens when it's light out. Rubisco's only active when it's light out, but there are times when we would need to see oxygen used as a substrate. All right, so just like we kind of had a glyoxylate cycle kind of activity, we'll have the same thing here for the chemistry that happens for photorespiration. And again, it's going to utilize some chemistry within the chloroplast, some chemistry within an organelle called the peroxisome, and then we have to dip just a little bit into the mitochondrion for some additional chemistry. So we'll spend some time kind of talking about this in our activity, but big picture is this happens when CO2 levels are depleted, and you need to have this enzyme doing something. So you have it use oxygen as a substrate. And as we'll see as we kind of go through here, this ends up being a very futile substrate or a futile cycle because you're basically going to use up a good bit of ATP and NADPH and you actually evolve CO2. So it kind of seems to undo a lot of the processes that work to a plant's benefit in photosynthesis. So clearly it has some benefit. And there's a hypothesis that that basically says if you don't have Rubisco do something, do some chemistry, there can be extensive photooxidative damage. And so you just need Rubisco to be doing something. And so if you allow it to use oxygen, it doesn't matter if you're using up ATP and NADPH, because if there's tons of light around, well, you probably have lots of ATP and NADPH to begin with. And the experimental evidence they have to support this hypothesis is that when they expose chloroplasts to intense illumination and they eliminate both oxygen and CO2, so there's no substrates available for Rubisco, the chloroplasts rapidly and irreversibly lose their ability to undergo photosynthesis. So photooxidative damage occurs. And so it's maybe kind of a protective measure. So the last thing I just kind of want to mention is, well, okay, if local CO2 levels become depleted, is there a way to kind of fix this? And from a man-made standpoint, there's really not. Genetic evolution or genetic engineering really hasn't been able to be very successful on this. And this can put a major limitation on a plant's ability to grow if you constantly have this futile cycle. But nature has a pretty cool fix with this, and this is this concept of kind of regulation in space. So I'm going to introduce some concepts. We're going to talk about C3 plants, C4 plants, and then CAM plants. So C3 plants are kind of what we've always been talking about. Calvin cycle produces 3 carbon carbohydrates. C4 cycles refer to the ability of plants to fix and store CO2 as a 4-carbon carboxylic acid, as malate. We've seen malate before. So what you kind of do is you take in and you store CO2 when it's readily available, and then you liberate it from malate as you need it to do chemistry. And what's necessary for this is a certain kind of leaf anatomy that allows you to have different kinds of cells. So we're going to see this, that you'll have these mesophyll cells, I'll flip to the next slide here, and these bundle sheath cells. So basically mesophyll cells lack Rubisco activity, but what they can do is take up and fix carbon dioxide, and they'll do this often at night. They condense it as bicarbonate, they combine it with phosphenyl pyruvate, they end up generating oxaloacetate, and then chemistry that we've seen in the citric acid cycle generate malate. So again, they generate malate, and they assimilate this CO2, but importantly, there's no Rubisco in these cells that are going to worry, about kind of utilizing that CO2 for other purposes. So you kind of reserve and store CO2 in these cells. As you need it, what you can do is you can export this malate to these bundle sheath cells, and then there'll be a special enzyme that can liberate CO2 from malate. You can decarboxylate it, and then that can be used within the Calvin cycle. So again, this is going to ensure that, and remember, the Calvin cycle is only active when it's light out, but it's going to ensure that you don't have to worry about depleted CO2 levels. And again, for certain plants which can't open up their stomata during the day a lot to bring in CO2, it allows them to kind of tap into a reserve of CO2 and still undergo the Calvin cycle chemistry. So again, C3 plants, what we spent our time for the most part talking about, fixing carbon dioxide as 3 carbon acids. So the majority of plants kind of do that. These C4 plants kind of live where it's very hot, very sunny climate. They can't, they readily will deplete CO2. So they kind of find a way to store CO2 at night so that they have it readily available during the day. So the last thing to kind of mention is these CAM plants, and when you kind of see these succulents, sometimes cactus is another kind of a CAM plant. It's a variant that basically separates CO2 acquisition in time rather than space, and I kind of hinted at this. So it's a special kind of C4 plant that, because they live in very arid, dry climates, really can't open their stomata during the day to actually even bring in CO2. So they open up their stomata at night, they bring in CO2 at night, but remember, we're not doing the Calvin cycle at night. So they fix this CO2 without light, storing it as malate, as that C4 reaction pathway intermediate, and then basically they can liberate it during the day and use it to undergo the dark reactions during the day. So that's it for photosynthesis. Pretty cool stuff, I think, especially as we find parallels with mitochondria.